This video is part 2 of chapter 5, and we will begin discussing proteins. Proteins are probably one of the most diverse macromolecules in living systems. They account for about 50% of the dry mass of most cells. Some of them speed up chemical reactions, and then there are many other functions of proteins which we'll get to in a little bit. They're all constructed from the same set of 20 amino acids. Amino acids are the monomers. We'll get to that in a minute as well. We call the polymer polypeptides. Peptide means protein, really. And we've talked about the prefix poly. A protein, though, is actually the biologically functional molecule that may consist of one or more polypeptides. As we mentioned earlier, the amino acids are the monomers of polypeptides or proteins. They all have the same basic structure, which includes an amino and a carboxyl group. But how they differ in their properties is due to the differing side chains, which are called R groups. This is the basic structure for all amino acids. You can see the amino group here, the carboxyl group here, which is also known as a carboxylic acid, so that's where the name comes from, amino acid. There's a central or alpha carbon, and then off of the alpha carbon is the R group. The R group can be as simple as a hydrogen, or it can be as complex as a carbon ring. And this the R group that distinguishes the 20 different amino acids from each other. When you string together amino acids, you get a polypeptide. And as we've already stated before, it's the structure of any molecule that leads to its function. Proteins exhibit more complex structure than the other molecules that we've looked at before. And we categorize protein structure into 4 levels: primary, secondary, tertiary, and quaternary. The primary structure is the sequence of amino acids, or put another way, the order in which they appear in the molecule. The secondary structure consists of the coils and folds in the polypeptide chain, which also includes interactions between the different amino acids. We'll get to the details of that in a second. The tertiary structure is essentially the 3-dimensional structure of the polypeptide. And then the quaternary structure results when 2 or more polypeptide chains are associated together. As I mentioned earlier, the primary structure of a protein is the sequence of amino acids, or the order in which they appear in the protein. It's sort of like the order of letters in a word, or the order of words in a sentence. This primary structure is determined by the inherited genetic information that a person possesses. Here we have an example of primary structure. You can see here, first of all, we've got the amino acids strung together. Notice that they are covalently bonded. We call this bond a peptide bond because peptide refers to proteins. But you see here, in order, we've got the abbreviations for different amino acids. There's gly, pro, thro, gly, and on we go. It's that specific sequence that determines the basic or primary structure of a protein. When the amino acids in the protein begin to interact with each other, we get secondary structure. And this typically results from hydrogen bonding between the different amino acids in the polypeptide backbone. There are 2 basic types of secondary structure. One is called an alpha helix, which is a coil, sort of like a spring, and then a folded structure called a beta pleated sheet, would look sort of like a folded accordion or drapes. Here's what those would look like. You can see the kind of spiral helical structure in the alpha helix. And then in the beta pleated sheet, you can see the kind of folded pleated structure. Notice the hydrogen bonds between adjacent amino acids in the polypeptide. Those are the bonds that contribute to secondary structure. If we add primary and secondary structure together, we get tertiary structure. This is a good example of emergent properties. The tertiary structure is the overall 3-dimensional shape of the polypeptide, and it typically results from interactions between the R groups more so than interactions between the amino acids themselves, that is to say the amino and the carboxyl groups. There are many types of interactions between the R groups, which include hydrogen bonds, ionic bonds, hydrophobic interactions, and a weak type of interaction called a van der Waals force. Oftentimes there are also covalent bonds that are formed between adjacent sulfurs, which might be found on different amino acids. These are called disulfide bridges. Here are some examples of interactions that lead to tertiary structure. Here's a hydrogen bond between R groups. Here's a disulfide bridge between R groups. Here's an ionic bond between R groups. So there's a number of different interactions. And then lastly, when you put more than one polypeptide together, you get what's called quaternary structure. Lots of proteins exhibit this, where you have multiple proteins wound together or interacting in some sort of way, either through bond, some sort of bond, or just other types of interaction. Collagen is a common fibrous protein found all over the human body, and it's coiled up like a rope. Hemoglobin is found in the red blood cells, and it's actually formed by two what are called alpha globin chains and two beta globin chains. Here's hemoglobin. You can see that there are two beta subunits bonded to two alpha subunits. So these are four separate polypeptides put together into one protein. The last macromolecule we'll look at is the nucleic acid. And these are really important for storing, transmitting, and expressing hereditary or genetic information. And as we mentioned earlier, the amino acid sequence of a polypeptide is determined by the unit of inheritance, which is called the gene. And a gene is made up of nucleic acids, primarily DNA, or deoxyribonucleic acid, which is one of the nucleic acids we'll look at. The monomers of the nucleic acids are called nucleotides. There are two types of nucleic acids, and we're just going to look at them in general here, and we'll get into more detail on these in a later chapter. DNA, or deoxyribonucleic acid, is the primary one, and then ribonucleic acid, or RNA, is the other one. DNA provides the backbone, basically, of the genetics of an organism. It's the blueprint for all the information in any given organism. It's also interesting in that it provides the directions for its own replication. We're going to talk about that later. It also directs the synthesis of messenger RNA, which is a particular type of RNA that controls protein synthesis. This process is called gene expression. We're going to spend a lot more time on that in a later chapter. As we mentioned earlier, the components of nucleic acids are nucleotides, so therefore the polymer is called a polynucleotide. Each nucleotide consists of three components: a nitrogenous base, that is to say a molecule made up of various nitrogen atoms and other atoms, a sugar, and then one or more phosphate groups. The portion of a nucleotide without the phosphate is called a nucleoside. There are two families of the bases, the pyrimidines and the purines. Pyrimidines have a single six-membered ring, and purines have a single-membered ring fused to a five-membered ring, so they have two rings. In DNA, the sugar is a deoxyribose, and it's a ribose sugar that has one fewer oxygen than the sugar ribose, which is found in RNA. Here's the basic structure of a nucleotide. Here we have our nitrogenous base, which could be a pyrimidine or a purine, the sugar, and then the phosphate attached to it. Over here we've got our different bases, cytosine, thymine, and uracil, which are the pyrimidines, and then adenine and guanine, which are the purines. And then here's the two sugars. You'll notice here's where the oxygen is missing on deoxyribose compared to ribose. And then you can see here the monomers are strung together to form the polymer. And the process that does that is dehydration synthesis, which we've seen before. DNA is actually two polynucleotides that are spiraled around each other, which forms what's called a double helix. The backbones run in opposite directions, which is based on the arrangement of the sugars and phosphate. We'll learn more about that later. And any given DNA molecule can include a number of different genes. Now, the way that the two strands of DNA attach is through bonding between the purines and the pyrimidines. And this is what we call complementary base pairing, where the bases hydrogen bond to each other. In a DNA molecule, A always pairs with T, or adenine always pairs with thymine, and guanine always pairs with thymine, or cytosine. This is what we call complementary base pairing. And this is the feature of DNA that makes it possible to copy itself prior to cell division. And we're going to spend a lot of time on all of these concepts in later chapters. RNA, by contrast, is a single-stranded molecule. Now, sometimes complementary pairing occurs, but that just depends on the type of RNA. Besides being single-stranded compared to double-stranded in DNA, One of the big differences in RNA is that instead of having the T or thymine, there's U or uracil. So uracil acts like thymine in that it will base pair to adenine. And while DNA always exists as a double helix, RNA molecules have more variable forms, and we're going to learn about those later. Here's a good example of a DNA molecule. You can see the strands wrapped around each other. Here's the base pairing, C to G, A to T. And then here's a type of RNA called a transfer RNA molecule, which has parts that are single-stranded and then some that are base paired together. And again, that's just based on the fact that this is a specific type of RNA molecule.